Becky.
Sure. Thank you, Meredith. Thank you all for joining today's call. I think most of you know about what IARPC, the Interagency Arctic Research Policy Committee, and this is the Marine Ecosystem Community of Practice. And IARPC, the goal is to, cross-fertilize discussions between the U.S. agencies interested in the Arctic, nonprofits, academics, and so forth. And the marine ecosystem team is basically looking at ecosystem structure and function in all aspects from interdisciplinary components to that and interactions with coastal communities also. And so with that brief introduction, I want to go straight forward to Katrin. Katrin Eichen is a professor at the University of Alaska Fairbanks. She is the lead for the Arctic Marine Biodiversity Observing Network and has been since the inception of this. And I have to disclose I am a co-PI. Gladly, we've been working with this team up in Fairbanks and other components. And she will be presenting the way things have been developing and some of the core results, the goals of the program. Katrin is a professor, like I said, and a marine biologist by training, and I think I don't need to go through your full academic background. But with that, I want to really welcome Katrin for being here and willing to cover all aspects of the AMBON project, which has been very successful, and it interacts with the actual program on the observing system that I'm involved in and many people that are on the line. All right, Katrin, it's yours.
Yeah, thank you so much. Let me share and make sure that everybody can see the presentation. You see it full?
Yep.
Okay. Sorry. Okay, well, thank you for the opportunity to talk about the AMBON project. And AMBON stands for the Arctic Marine Biodiversity Observing Network. And we obviously do biodiversity observations, and we affectionately call it from microbes to whales. But before I go into an update of what we've been up to over the last few years, I'd like to acknowledge the team. This was definitely a team effort, as well as our funding agencies, particularly BOEM, NOAA, NASA, and ONR, but there also was supplemental funding from AUS and NSF. So our goals were to provide high quality biodiversity data for the Chukchi Sea, I guess, again, across trophic levels. And the work was focused on both new data collection, but also on synthesis of existing data. And AMBON is part of the National AMBON initiative and provides a Arctic perspective to that initiative. We are committed to open access data management, and that is for this audience, other academics, but also particularly other stakeholders such as management agencies, and we're aiming to provide useful and actionable stakeholder information. So I said working across trophic levels, so I couldn't resist in showing this little cute image of the food web. So I will talk about a number of these elements of the what the AMBON project did, but I will not have time to talk about everything. So before we get going, why the Chukchi Sea? And I know in part I'm probably preaching to the choir here, but it is a highly productive Arctic shelf system. It experiences one of the highest rates of climate change. It also has a number of sentinel species, such as many birds and marine mammals, many of which also are of subsistence harvest interest. Well, currently there's no commercial fisheries in the Arctic. We do have an Arctic fisheries management plan for the potential of the fisheries in the future. And so the data that we collect and provide contribute to the informing this management plan. It's a region of increased vessel traffic, and while there is not currently any oil and gas extraction on the Chukchi shelf, there are lease areas, and so again, there is a potential for future activities. Accessibility due to ice cover and high cost of operating in the Arctic are problems that every project in the Arctic is facing. And so we were partly aiming to develop a sustainable approach for doing this work. And this was mainly through collaboration with other programs active in the Arctic. And Jackie just mentioned the DBO, the Distributed Biological Observatory, which was one of our main partners in this. You see here on this map, you see these DBO lines. I believe that probably everybody on this call has heard about DBO and the DBO lines before, as well as the CEO located here in the northeastern Chukchi Sea. It's the Chukchi Ecosystem Observatory. So those were the two main partners, and I will refer back to those here and there. We also aimed to work with NOAA's Eco-Foci program, and just based on logistical things, it didn't quite work out during sort of this project, but we have built up on that collaboration in more recent years. Okay, so starting out with field work in the project period that you see here, which actually included some no-cost extensions, so the actual field work was much shorter. If you look at the dates, then the first thing that comes to mind is, yes, those were COVID years. And that caused a lot of logistical fieldwork problems that I'm not going to go into, but we were in the end able to launch two field seasons, one in October 2020 on the Norseman 2 and one in November 2021 on the Sequoia. And at first we were a little hesitant about how data from this late in the season would be comparable to data that were collected typically in previous years, much earlier, more towards August. However, it turned out that this was really quite interesting, and we learned a lot about the system during these late year collections. So I'm just going to zoom and basically fly through some of the results that we found across these trophic levels. These are data from October 2020 on phytoplankton. The data actually displayed here are integrated water column chlorophyll data. And again, across these DBO lines. And you can see here how there are some regions of really high, for us, unexpectedly high phytoplankton biomass this late in the season. And for this region here, you can also see some plots on nutrient availability, and you'll see very high nutrient availability in these a little bit deeper waters. But these sort of high phytoplankton biomass this late in the season was a bit of a surprise. Another aspect of what we did during the reporting period here was that we used data from the CEO sediment trap, and which collects sedimenting material over the course of a year. And so we supported some work looking at the samples. And here you see basically a time series of these various colors are different diatom species. And in yellow, you see sunlight availability, as well as in gray sea ice. And you can see that, well, as expected, overall diatom abundance is higher in open water than during ice cover, but we can see that different species have fairly high adaptations to certain periods of the year. You know, some are really open water, others are peaking more towards the onset of sea ice covers. Others start to peak early in the year when there's still quite a bit of sea ice cover, but sunlight is starting to come up, and then others are really more peaking during the breakup season. So we get a much more refined picture by looking at sort of year-round data in collaboration with the CEO. In terms of zooplankton, I'm showing you here just some species abundances for the 20 and 21 late season collections, and then in comparison to some collections that happened in earlier years during earlier timeframes. And you can see that some species such as Calanus, the typical high rich species, was very low abundance late in the season compared to earlier seasons. And it's likely that this species has started at this time to go into diapause, so goes into deeper waters off the shelf, while the more shelf-dwelling species, Pseudocalanus, remains relatively high in the fall. And then maybe particularly interesting was the high abundance of 2 krill species in this late of the year, much higher than during the summer. And that is important if we think that species such as bowhead whales do their fall migration. So this could be a really good food supply for these whales while they are transitioning through the Chukchi Sea. Moving on to the benthos, this is work that Jackie is doing, and again in collaboration with the DBO. And so these are data from October 2020, and we do see relatively high abundances as well as in places biomass of the macrofauna. And these patterns are actually not that different from what we see in during earlier seasons in other years. And it's likely that the longevity of the benthos is sort of buffering these seasonal changes. So if they persist through many years, then we wouldn't expect necessarily sharp declines during the fall and then into the winter. Why do we see some areas that have really high abundance and biomass, while others have really high biomass but low abundance? That is related not to the season. This is related to which species or which general taxa we find there. So in these regions where you have both high abundance and biomass, they are dominated by clams, which are heavy. They provide a lot of biomass, while these areas that have really high abundance but low biomass are more dominated by smaller things such as amphipods and worms that just don't provide as much biomass. Now, one still needs to think about how do they do this? How do they persist through the year? And so we can link this information to actually sediment information from the same year, from 2020. And here you see sediment chlorophyll A and sediment total organic carbon. And we do see fairly high amounts of these metrics in the sediment in October. And so this means that there is a lot of food that accumulates during the peak productivity time and then sinks to the bottom and gets incorporated into the sediment and provides what we consider a longer-term food source or a food bank for these benthic organisms, which assists them to basically persist through the year. In terms of epibenthos, so the critters that live on top of the sediment, we were not able to collect any new data, but we used this opportunity to do a data synthesis. This is also in collaboration with a NPRB project. So we combined forces and did a data synthesis between 2009 and 2024 using a vast model approach. This is a vector autoregressive spatial temporal model. And so we modeled the biomass based on the collections happening during those years. And here in this overarching or in this summary figure, you see the modeled biomass over years. And what we can see is that there was a high biomass in 2009 and 10, and then there was a really sharp decline after 2010. And then from then on, biomass remained fairly low with some variation. And if we look at these individual year maps, we can see where these declines happened the most. So in these maps, light colors, yellow colors indicate high biomass, and blue, dark blue colors indicate low biomass. And so you can see that particularly in the northern study region here, we had these really high biomass in 2009 and 10. And so if you look in later years, that's where we see a lot of the decline, but pretty much across the board. But we can now start looking at correlations with environmental variables that may have changed in these various study regions as well that might be related to some of these declines. For the epibenthos, we also looked at diversity, and we took a diversity index that's called delta, that takes taxonomic relatedness of the various species into account, as well as their contribution in biomass to the total community. And what you see here are, again, these individual year maps. Now this is modeled diversity, delta diversity, with yellow colors indicating high diversity and blue colors indicating low diversity. And what we see was a surprise at first, where we see an increase in diversity over time. And you can see this here. Some regions remained dark blue. There was very little change, and others changed more strongly in diversity. So we dug a little bit more into it. Is it an increase in species? Is it an increase in potentially boreal species? But what we found was something different. What we found is that it is related to a decline in particularly abundant species, and that was particularly snow crab and brittle star. So we saw a fairly sharp decline of these two taxa. And when you go back to what I told you, it takes the taxonomic relatedness, but also the contribution of the taxa in biomass into account. And so if you take away the big biomass dominant species, you get a more equal distribution of species, and that in the index actually leads to a greater taxonomic diversity. So similarly for fish, we were not able to collect new data, but we did a synthesis, this time looking at functional diversity. So this is a synthesis from 2007 and to 2019, and it's looking at available fish community data, but analyzing them based on functional traits, not on their taxonomic identification. And so the functional traits that we took into account was age at maturity, fecundity, longevity, body shape, life stage, and body size. And when we characterize the community according to those functional traits and then do a cluster analysis, we find three main clusters: a blue one up here that we call a northern cluster, a green one that's sort of here, and an intermediate cluster, and then a red one that goes all the way down here, which we call a southern cluster. And then you can look at how these different clusters are characterized in terms of their functions. And what it can do is that we can now look at what drives certain functions in terms of the environmental conditions, but then also we can look at what might make new species that might invade a particularly successful competitor. Moving on to marine mammals, one way we looked at marine mammals was through passive acoustics, which the AMBON project supports as part of the CEO mooring array. And so we've done this over a number of years now. And so here you see a compilation from 2016 to 2020. And this is just the data for walrus and bearded seals. And what you see here is, again, time. And these are the vocalizations from the respective species. And in orange, you see sea ice cover. And in these little red bars just indicate that there were some data lost because of malfunction of the hydrophone. And what we can see if we look at these two species that are both abundant in the northern or northeastern Chukchi Sea Hana Shoal region, we can see some habitat petitioning. We see that walrus vocalize a lot more during either low ice cover or broken ice cover and open water period. You can see this across all years. Then during the really heavy ice cover, while bearded seals do the exact opposite. They vocalize mostly during the heavy ice cover and much less in the open water period. So again, seasonal habitat petitioning between these two species. A different way that we were looking at marine mammals was through eDNA collections. And so this is from data that we collected, or samples that we collected in 2021. And here you see eDNA is environmental DNA, where we took water samples from the CTD and then analyzed the water sample for free DNA that is from species that are continuously shedding cells or DNA into the water. We analyzed these data or these samples for a number of different primers that target bacteria, archaea, invertebrates, fish, and marine mammals. And I'm only showing you the marine mammal data. And you can see here that while maybe not particularly abundant everywhere, we were able to see or to detect a number of different cetaceans. We also detected pinnipeds, but I'm only showing you the cetaceans. And you can see, for instance, beluga whales were particularly abundant, as well as gray whales. And so this was a nice proof of concept, which we're still refining as of right now, that we can use eDNA for marine mammal detection. So I mentioned in the beginning that we are aiming to provide actionable information for stakeholders. And so I wanted to give you a couple of examples for this. So for instance, some of our team members are part of the North Pacific Fisheries Management Council Scientific and Statistical Committee. Sorry, it's a bit of a mouthful. And so basically being able to provide statistically sound advice and scientific data to fisheries management is something where the AMBON project is able to provide sort of an Arctic perspective to what's going on also in the Bering Sea. We are aiming to assist with management agency needs. So, for instance, we have representation on the National Academy of Sciences Committee, on Offshore Science and Assessment, the COSA Committee, which is a committee that supports BOEM in decisions on their environmental studies plan, or provides feedback on environmental study plan, as well as other scientific endeavors, as well as that the information we provide is useful for spatial planning, following invasive species, NEPA assessments, and such things. We also, several of our team members were part of the Arctic Integrated Ecosystem Assessment of the Northern Bering Sea and Chukchi Sea. That was a Pisces organized working group 44 that completed its work just last year, and so completed during the AMBON project duration and submitted a report on this. And then some of our team members have been very active also in international Arctic science planning, for instance, through the Arctic Science Summit Week, where they organized specific town hall meetings on specific areas or interest matters, such as the UN Decade of Ocean Science for Sustainable Development at the Arctic Science Summit Week in 2022, and then another one in 2023 towards a Goose Regional Alliance for the Arctic. There are other examples, such as the PAG group and things like this, but I just wanted to give a couple of highlights here. So what are the next steps? We actually have a follow-on AMBON project that is funded by NASA and ONR that is currently ongoing. And here we are focusing on combining and linking different observations. We have annual vessel-based surveys through the based out of Sikuliaq. And we take a lot of samples from CTD, and part of that hydrographic data, but part of that is also a focus on eDNA collections, and also on benthos and fish collections. Then again, continuing long-term monitoring with the CEO, where we are aiming or we're working on looking at eDNA collections from the continuous water samples. So getting eDNA samples throughout the year. Particular focus again is on marine mammals. We continue to support the marine mammal passive acoustics. And we also have a benthic camera that was originally supported during the AMBON project that I'm reporting on today through additional funding from AUS. And so now we just last year got our first set of images back, and it's super exciting. Hopefully another time I can talk a little bit more about that. And really exciting is a new collaboration that we have through partnership with the Alaska Arctic Observatory and Knowledge Hub, AOK program, with the native village of Kotzebue, where we visited with the community, and they have a very great interest in also collecting eDNA data. So we provided them with materials and instrumentation for this, and we're supporting collections of that, as well as of coastal observations through citizen science. And then we analyze the samples and report back to the community. And again, so this AMBON 3 phase is again a partnership with the DBO, with the CEO, and at this time much more intensively also with the EcoFoci program. And with that, I think I'm probably a little over, but I wanted to thank you for your attention, and I'm happy to answer any questions.
All right, thank you, Katrin. Your timing is just fine. You know, we have a lot of time for questions, so I'm putting that forward. I have a question or just a comment I would say is that you're interesting about your biodiversity in the Delta part. I mean, this is it. We're seeing changes in the macrofauna biodiversity, and where we have those real hot spots, a high biomass, low diversity, you know, that change is causing a drive down of predator prey. So it's kind of nice to do that comparison with the epifauna. So that's really interesting data. So
yeah, absolutely. And there's a lot that we can now delve in a little deeper in terms of, you know, the more on an ecosystem function perspective. What do crabs do to the system? What do brittle stars do to the system? If you now have lower biomass or abundances of those, what does that mean? You know?
Right. No, I think it's a really it's exciting, interesting data findings. So are there any questions? So people can raise their hands, or they can put it in I'm checking the chat box and I don't see anything yet. Anything you want to ask Katrin? Let's see. Not yet. Let's see.
Christina.
Hey, Katrin. I'm curious. So you obviously a ton of interesting results from all this. And with sort of the next version of AMBON ongoing for you, and maybe this is a question for Jackie too, hearing from both of you, are there any specific like questions or threads of research that you're interested in following from this past work?
Yeah, so I'll start, and then Jackie might chime in on this too. A little bit of what I touched on in the talk here. eDNA is a very strong component of our new AMBON, and in part this is based on the initial findings that we did through what we call AMBON 2. It's just a little easier, which is what I reported on here. So some of the, you know, just initial proof of concept, for instance, detection of marine mammals, which is something that our ONR funder is extremely interested in. That is, and also the native village of Kotzebue is very interested in this as well. So basically that was set up through that AMBON 2 and is now being carried forward into AMBON 3 as sort of a major focus. The other thing is, and that is a little bit what Jackie also just said, is really following more onto the biodiversity synthesis data. And that is both true for epibenthos as well as fish, and potentially also for the macrobenthos, to really look at this in more detail, A, at what is driving this, but B is what does that mean? Again, at least for the epibenthos so far, there's no glaring influence of boreal species. So the topic of borealization that you find oftentimes in zooplankton or fish larvae or even adult fish and things like this. We don't see this quite yet so much for the epibenthos, but the changes we are seeing are already, they're likely to have, consequences for certain processes such as bioturbation, respiration in the benthos, those kind of things. So those are some things that we're digging in deeper now during this AMBON 3 phase. And just sort of as a quick glance forward, the benthic camera that provided us with near year-round data or images is already telling us a lot about certain species behaviors that we will probably be able to link to certain ecosystem functions, particularly in the realm of bioturbation. So again, that was all set up, and then we're carrying some of that forward.
Yeah, I would just add that I think that, one of the driving things we're looking at is the habitat changes. I mean, the environmental drivers that are causing these changes, and even for the biodiversity aspects, for example, the changes in biomass that really tie in for upper trophic animals. So, we actually have there's different modeling projects within the observing system and it crosses over with the AMBON, with the DBO. But we're also looking at potential of a new postdoc I have looking at operational aspects. Basically, where would you, timing of impacts of changes in like migration corridors and what impact that might have. And that's all really tied to predator-prey activity, right? So you change the food base. These animals are not going necessarily after one species. They're going after something nutrient-rich on the cafeteria, and so they will change. And can we even get to the point where we could forecast biology? That's always a big question. But I think that the beauty of the AMBON project is the fact that we network at different programs that are looking at a lot more than any one program can do.
And then one other thing that I didn't even touch on here, but that we worked on during the AMBON 2 phase is seascapes. And I just didn't have time. I already talked a little longer than I was supposed to, and I just didn't have time to touch on this. But now that you give me an opening, Christina, I can at least mention it. So seascapes is a remotely sensed habitat classification, and we worked during AMBON 2 on this because this is a global product, but for the Arctic there are some problems, particularly in shoulder seasons and during ice cover and things like this. So some of the typically remotely sensed variables are not all that reliable. And through some additional EOS funding during AMBON 2, we developed some new approaches to this, where we focus on a regional model instead of the global seascape model that uses some of the PAROMS data, and particularly Kate Hedstrom and Seth Danielson have developed. And we're even using a physics-only model that is only using some physical data. And so we have sort of this regional model, which is quite reliable. And we're working towards now using the seascape model that was developed during AMBON 2 and try to link it to some of the biological data during AMBON 3.
Great. I noticed this, oh, here we go. Great, let's see. We have time series data on ostracods. So Laura Gemmer has something in the chat box, basically an information about the ostracods, which are bivalve crustaceans. Since AMBON has funding
Yes.
through 20, would you be interested in including the group's abundance and diversity data in the AMBON accounting? And,
yes, absolutely. And Laura, I'm owing you a response. We communicated a little bit about this already, and I wanted to make sure, and we talked about also, for instance, collecting some more samples for you, but I needed to confirm this, and we just had, 2 weeks ago, we had a meeting, a cruise planning meeting, and I did confirm that yes, we can and we will, and so I will get back to you. But of course, I mean, providing you with new samples, but also bringing you more into the fold of yes, these biodiversity assessments and adding another element to that. So sorry that I haven't gotten back to you yet on more of this.
Yeah, I would just say we collect for Laura, on
Yep.
surface sediments if you want, but so
Yeah,
she's
and
been-
I maybe it was you or it was Lee. I don't remember with whom I talked, both of you.
Yeah, so she's been finding seeing borealization aspects and finding new species, particularly in the Alaska
Yes.
coastal water sediments. So I think it's looking at all the different trophic levels is a real valuable thing, and obviously in the AMBON network on that.
Yes, absolutely.
Yeah, and the only other thing I was going to add, while somebody asked for more questions, I think the bentha camera was brought up, and Christina mentioned that, and I think that the pictures of seeing that crab get rid of its shell and grow big is worth 1,000 words, and I think
Absolutely.
because I think you had about 10,000
8,500 images.
Okay.
We're not entirely through the year for some reason. We don't know. We deployed in, I want to say, August or early September, and it only started taking images in October. We have no idea why it didn't take any images for a month, and then all of a sudden woke up and started doing its job. So there's some glitches that we're actually working with the company trying to figure out what is going on. But 8,500 pictures, lots of them in December, January, February, March, sort of that time of year where we really don't know much what's going on. And so yeah, one really like the showstopper of this whole thing is that we had in January a crab, a snow crab, move into the picture frame, which is a little bit more than a meter squared seafloor, and it moved around for a couple hours and started to molt. And you can see the steps of molting of the crab. The sort of the back end comes out. It's a different color and everything. And then it molts, and then you can see how the molt, the empty shell, the molt, is much smaller than the crab that comes out, which is the whole purpose of molting. And being able to see this, catching that on camera, that was just super cool. And then the crab just moves away and lives its happy life, we hope. But the mold just stayed in that picture for, I want to say, 17 more days. And you can see all these little scavengers coming and feeding on it. And it's just, there's a lot of interesting observations that we had. We had a dead jellyfish move through, and you can see all the brittle stars moving in on it, and also some behaviors of brittle stars, and just the amount of other critters that just zoom through, and the speed at which they move. Those are all things we can now look at, in addition to some of the co-located physical data, such as which wave height might be one that then actually has impacts on the benthos. What storm surge do you need to stir up the benthos, because it's located with the CEO, so we have all the data for this. So we're actually working on a proposal right now to get some funding to analyze all of this.
That's great. And so do you see the seasonal suspended load variance then through?
Yes.
Yeah.
Yes.
Okay. Yeah, I would imagine so, and that has an impact on settling.
Yeah. Yes, exactly. And so the images are taken, when they are taken, every 30 minutes. So it's a reasonably high frequency.
Okay. And then
I'll go on. If you don't interrupt me, I'll go on this camera for an hour because it's so cool.
Yeah, I was going to ask Lyle a question because I know he's still online, but before, do you see fish going by on yours?
We do. Yes,
we have a drop camera we do in July.
Yes.
And definitely during the time in, the 18 and 19 period when there was no sea ice, production, then we started to see pollock and cod even on our camera, and krill swarming on the bottom. So I was just wondering if she saw these events too.
We do fish reasonably regularly come through. We do see shrimp. We don't see krill really all that much. But then again, I have to admit that I haven't looked at all 8,500 pictures yet. So I kind of, clicked through. but we definitely see sort of that more epi or suprabenthic krill, shrimp, Eualus, that's very abundant, and we see those come through all the time.
Okay. Yeah, and I was asking Lyle, I see you're on there. If you had any comments about the the fish slide that she had, or the diversity issues, I was just curious in relation to the work you're doing.
Well, I'm just really glad to see that she's getting it. I mean, we always think that, particularly when you start getting to particularly some of the larger fishes, that the spatial demographics sometimes won't work to get that. So it's nice to see that data is showing up and that we can maybe try to work with it.
Yep.
We haven't spent as much time doing benthic fish work in the Chukchi Sea. I mean, we haven't been back since 2012. So, yeah, I mean, I think this has great value and has great utility for us, and it would actually help us to align. And I think once we get to where, there's a lot of interest in having, say, the bottom trawl survey happen again in the, particularly the southern Chukchi Sea, but really all over in the Chukchi Sea. This kind of data actually gives us the ability to do like interannual modeling between survey events that could have a real powerful impact.
Yeah, and Lyle, this is so I want to say that NOAA data are included in this data set, and this is based on a data rescue effort by Franz Mueller and Jen Marsh. That basically put all the information together, and then we took that data set and analyzed it for the functional composition. And there is, we also, the paper that is actually fairly advanced, so I think probably in the next months we'll submit this for publication, also included a taxonomic component. So doing the cluster analysis and the modeling and the ordination and ordinations in relation to the environment, which none of which I showed you because again I had no time. We did this for the functional approach and the taxonomic approach in comparison, and so that's very cool because what the taxonomic analysis did, it picked up a nearshore component or a nearshore cluster, which the functional did not. Which is that's one aspect to keep in mind. But the other is that the functional clusters were way more clearly separated than the taxonomic clusters. And so I think in combination, those two can be super informative.
Yeah, actually, I was the one that provided a lot of the data to Franz and Jen when they were working on this. And so I'm very familiar with a lot of that work. I guess I took Jackie's question to be more about your kind of more recent imagery and kind of what we can do with that. And I'm kind of excited about that because we don't have a lot of new data. So
yeah. And it's definitely sculpins, I want to say eel pouts, Arctic cod that we see. So for those, I will need a little bit more help. I'm not a fish expert, so some of them it's like, I think that's some sort of a sculpin, but
yeah, well, that's great. No, that's it. It's that interconnection that's really, I think, really, really strong. Does anybody else have any further questions? I mean, I could ask questions, but I know Katrin, and we have the opportunity to do this. She is going to lead a cruise. She's chief scientist for our upcoming cruise. On the Sikuliak, and so that'll happen in August and September. So she'll be chief scientist on that, and we'll be collecting more of the state and hopefully bringing back both camera systems, right?
Yes, we still have a camera system that's out there that went away from us. So we're bringing an ROV to actually find it and bring it up. So ideally, we bring 2 camera systems up this summer. That would be a wealth of data that will be hard to manage, but potentially some very, very interesting data.
Yeah.
And
I would just say that
I see Gay is online. Gay, the cruise is, again, you probably know this, but it's from Nome to Nome again. And so we'll be in touch again about doing a straight science talk when we come back. We discussed a little bit about would it be better to do one before than after, because then you could, but I think it's really cool to have one after where we do hot off the press kind of reporting.
Did you hear
Or
what she
both.
said, or both?
And I say that actually, Katrin, because that's how Sekuliak's done it in the past as well. And it kind of gives people a background before you come back and sort of throw it all. Just saying. And that was going to be my question. So Jackie, you
we'll
got
think
it, or
about
you
it
already
and
saw my name.
see what we can do.
Yeah, thanks.
All right. Are there any other questions? I don't see any in the chat, because then we can do just final community updates. I want to thank you. Go ahead, Gabe.
I guess my last question is, does anyone have a prediction on what the ice is going to do this year? Because we are all over the map.
No, I hear you got,
It's going to be very interesting.
yeah. Well, you had ice go away, your fast ice went away, now it's come back, you know.
We were at like an ice minimum, like it was
Right.
2018 for, you know, we were all bracing ourselves for what will happen with that, with the ecosystem. And now we've had, I mean, I guess prayer works, because everyone around in this region has been hoping for north wind, and that's all we've been getting is north wind. So the ice is moving back down, but it's in poor condition. And I'm meaning it's really unstable, really mobile, crushed up, and sort of seems rather wind driven at this point. So it's piling up on the north shore of St. Lawrence Island. It's kind of emptied out of eastern Norton Sound quite a bit. So I'm just wondering, because we are going to dip down into the colder temperatures this week, and then we don't know what happens next. But if anyone has any thoughts on that, we'd love to hear them, because I don't know if it's enough last minute to make the cold pool robust, but and we are going to have to suffer if it's not. You know, we've been through this drill for the up and down the coast for what it means for our communities here and the ecosystem and so forth, so on. Okay. So when you guys get that ironed out, the timing of ice, what we need to make a robust cold pool, and how much algae grows quickly on stable ice or unstable ice, whoo, that'll help too. Make a prediction about what we may see come springtime in the future.
Okay. Yeah. Well, there's the challenge for everything from all the trophics to the modelers on that.
Well, we're so interested in what you guys are finding, because it's a matter of it's a strong matter for everybody on both sides of the strait at this point.
Yeah, and one of the things also, you know, there's collections of harmful algal bloom measurements that are done on these annual cruises, and the collections that Katrin and I have all collecting animals put into that network. So I think that is another thing that comes that for the food security topics.
Yeah, it's about $1 an egg right now.
Oh wow,
Okay.
yeah. We're
Katrina,
catching up, but I'm not that. We're at like $7 a dozen. But anyway, so yeah, so thank you, Katrin, very much. Before we close out. Oh, Doug. Okay, sorry.
I have a really quick question, Katrin, and I think I know the answer. Kathy Coolitz is not on. These cruises, is the seabird observations that were done. They're linked to this, right?
Correct. And seabirds was another aspect I didn't really talk about.
No,
again,
it's
because,
fine. Okay, we'll do it later.
Yeah, we did have collections that the AMBON project supported. I think it was the 2020 and 2020.
Yeah.
It wasn't quite co-located with the other cruise that we did, and that was all COVID related mess ups, but we did support those collections. And then we also did another project that is looking at shearwaters and collaborating with Australian colleagues on, they have geolocators on the shearwaters, and we used stable isotopes to identify any kind of, if we can link them to the foraging grounds in the summer. And interestingly enough, those were breeding birds, and none of them actually ventured into the Chukchi Sea. That was a surprise.
One of the reasons I'm asking you is that I didn't realize until your presentation that AMBON was I thought it was mostly Chukchi, but I see it's Northern Bering Sea. And Jackie, this relates to what we were talking about yesterday.
Yes.
Katrin, I'll be talking to you later.
Okay, that sounds good.
Yeah, I'll just say, Doug, yeah, we do have with Liz Lubutsky taking over.
Yeah.
She's been fighting to keep the at sea program going, and we have confirmed Fish and Wildlife supported seabird obs in both our observing system in July on the DBO and the Laurier Canadian cruise, and also in August, September on the Sikuliak. So the seabird observations at sea are a go. Okay. All right. So thanks. So any community updates? We have like 7 or 8 minutes. I would just say Arctic Science Summit Week is coming in Boulder next week. And there will be presentations, and I'm going to talk to you, Katrin, after this, because I'd like if you let me take some of your slides so I can give it as part of the observing the both the U.S. update through the Pacific Arctic Group. I'll be presenting there, but also the DBO. We have a community meeting, so I would say, but all different aspects of planning for the International Conference Arctic Research Planning. That's the science part of Arctic Science Summit Week, but the first 4 days are all the business part of it. So just to let you know, that's going on. So does anybody else have any community updates that they want to bring forward? I'll just add another one while you think about it, is that there will be the ESAR, I think it's ESAR 8, which is International Study Arctic Research for the Japanese program. They have a new icebreaker coming online. They want to have an international program. That'll be the end of September, a meeting in Tokyo. Okay, Gay?
Jackie, I just threw it. I don't know. You said community announcements. Otherwise, I wasn't going to put it in, but I just put in the chat. Rick Toman's doing Thursday night straight science, 6:30 p.m. Alaska time. And he's talking about the timing of our sea ice changes. He's focusing in on Nome. I think there's a bigger project he's a part of with some of the social economic issues. But there's been a lot of interest from some people locally that have been reaching out to him about a lot of the freezing rain events we're getting. So I think he might touch on some of our, maybe bring us up to date where we are.
Oh, excellent.
It's not showing up in the chat, Gay. Did you hit submit?
Yeah. Oh, here we go. I guess I got to push a button.
Oh, there you go.
Thank you.
All right.
Perfect.
Thank you.
So, there any other announcements or comments?
Yeah, sorry if I missed this and you already said this, Jackie, but I'm putting a link in the chat to Gravel to Gravel initiative, joint meeting of the terrestrial, marine, and coastal resilience teams on March 20th.
No, that's great, Melissa. I hadn't mentioned that, but it is the connectivity for land and salmon issues and others that are being, so we're cross-linking the marine ecosystem team that we're kind of having the meeting now with coastal community, be it ecosystem related topics like Meredith was just saying, or coastal community interactions, needs and suggestions for research. So that's great. So that's one thing to keep in mind. And if you have topics, please don't hesitate to send them to any one of the leads. Lyle is a co-lead, Danielle Dixon and myself, or to Meredith or Tony to about topics you'd like to have on these monthly webinars that we have, which is the second Wednesday at 2 p.m. Oh, here's another meeting of interest. What's that one on?
That is, sorry, that's the the modeling and observing ocean waves meeting that the physical oceanography team is hosting. And then here is a link to a form where you can suggest meeting topics. Yeah.
Oh, good. Okay. Thanks. All right. If there are any other comments or suggestions, ideas, updates, I can't think of anything right now. I know there's more meetings coming along the pipeline, but they don't come to mind right now. So with that, I want to again thank all of you for your, Gay, you want to say?
No, I just said just want to say thank you, Jackie.
Yes. And Katrin, once again, thank you for the time and everybody on with great questions on this, and Meredith for setting this all up. And with that, I'd say I give you back a minute or two since I have another meeting at 3.
Okay, great. Thanks, everyone.
All right. Thanks a lot.
Thanks again for having me.
You bet. Bye-bye.